Okay. Uh, Hello everyone. Uh, I will introduce education and research in medical cell biology. There are three staff, Hino and Koga and me, Nakao. Our activity um, for education and medical science. This slide shows overview of research. Uh, Environmental factors like nutrition and inflammation and stresses concentrated into nucleus for gene control. So basically transcription uh, induce uh, immediate response. However, under long-term or repetitive environmental factors, so epigenome is fixed. We call epigenetic transcriptional memory for adapted response. So resulting in health and constitution, lifestyle disease, and aging. So what is the epigenetic regulation here? So we think epigenome has three layers. So linear epigenome by DNA methylation and histone modification, and chromatin loop formation, and then nuclear domain uh, transcriptionally active genes make transcription factory, like here, and inactive genes make heterochromatin. Among them, we focused on methylation because this is stable chemical modification. For example, cytosine methylation and DNA methylation, and also protein methylation like lysine methylation. So we have several projects, and first is regulation of metabolism by LSD1 family. So lysine specific demethylases LSD1 and LSD2 repress metabolic genes by H3K4 demethylation. So LSD1 co-regulates cell type and metabolic memory in these multiple tissue types. We are analyzing this metabolic memory. What is a metabolic memory? Second is inflammatory memory in innate immunity. There are two types memory, trained immunity and also endotoxin tolerance. The first stimulation and second stimulation. We interesting in the second response. So in this case, you can see enhanced immunological response. In contrast, you can see suppressed response here. So enhancement and suppression by inflammatory memory. So we are focused on another lysine-specific demethylase, KDM7, for immunological response and inflammatory memory. Third is analysis of epigenetic factors that protects senescence. So epigenome and nucleolus and ribosome and also mitochondria, these three cooperatively function in the cells. So we screen the epigenetic factor protect senescence. Now we have about 20 candidates. So these candidates are involved in cellular senescence, actually protect senescence. We can understand what is aging epigenome. The novelty is phenotypic variation induced by interaction between environment and epigenome. So you can see genotype A and produce a healthy phenotype, and genotype B cause disease-prone phenotype. So under environment and epigenome, so you can see this variation. So genotype A have this variation. Also genotype B is a variation. So you can see in genotype A, you can see a few disease prone. However, genotype B has a few healthy population. So this phenotypic variation is very important for our health and disease by epigenetic regulation. So summary, current research project, and the basic part is the mechanism of epigenetic gene regulation and metabolic memory, inflammatory memory, and aging epigenome, and also for metabolism and therapy resistance in cancer cells. There are 3 books from our laboratory here. So please let me know if you have any interest. This is my email address. Thank you very much for your attention.